Hello, bachcho. Let's see the question. So the question says polymerase chain reaction technology, that is PCR, is used for. So we need to identify out of the four options where PCR is actually used. So the key concept in this question is to firstly understand what is polymerase chain reaction. So polymerase chain reaction, which is in short known as PCR, is a very important laboratory technique which is used for several purposes. So PCR is used to actually make copies, that is around millions or billions of copies of a specific region of DNA, which means that PCR is used for DNA amplification. So we can use PCR to amplify or to make several copies of a particular region of DNA, or we can say that it is used for DNA amplification of the gene of our own interest. So basically the main goal of PCR is to make enough copies of gene of our interest so that this can be used for several other purposes, like for DNA sequencing or for genetic engineering. Because working with a very low quantity of DNA is difficult, therefore we can make several copies of a single DNA template and then use it for various purposes. So this is the main goal of polymerase chain reaction. Now, if we go through the basic steps of polymerase chain reaction, then firstly in the PCR machine, the original DNA whose copies we want to make is added along with a DNA primer, which is particularly designed for this template DNA, and certain nucleotide bases are added in the sample. Along with that, a very specific enzyme, which is known as Taq polymerase, is added along with it. Now, this enzyme is specifically used for PCR reaction, and Taq polymerase is an enzyme which is derived from Thermophilus aquaticus, which is a bacteria. And the special feature about Taq polymerase is that it can withstand very high temperatures of around 96 degree centigrade. So Taq polymerase is actually a DNA polymerase which can withstand very high temperatures. Now the first step of PCR is denaturation. So these steps occur at around 96 degree centigrade. So at such a high temperature, the two strands of DNA separate. And along with it, the Taq polymerase remains intact because it can withstand this high temperature. This is the reason why we use Taq polymerase and we do not use any normal DNA polymerase. Now, the second step of PCR is annealing. So the primers then anneal to the specific DNA region. So the primers are designed in such a way that it will go and bind to the DNA sequence which is of our own interest. So the primer will not bind somewhere else, but it will bind specifically to the gene of our interest. So the annealing will occur. Along with that, the Taq polymerase enzyme will also attach to this hybrid, and this Taq polymerase enzyme will then help in the next step, which is extension. So the primer will then extend by addition of the nucleotides to the primer, which is complementary to the parent template strand. In this way, we will get two copies of the single parent template DNA. And when this cycle is repeated for several times, then this is how we get multiple copies of the same DNA strand. So we get multiple copies by repeating the cycle. So this is how PCR reaction takes place. Now, if we come back to our question, then the first option says that polymerase chain reaction is utilized for DNA identification. So this is the wrong statement because PCR is not utilized for DNA identification. The second option says DNA repair. Again, PCR is not used for DNA repair. The third option says DNA amplification. So this is the right option because we saw that PCR is used to make several or multiple copies of a single DNA strand, and therefore it is used for DNA amplification. So this is our correct answer. And the fourth option says cleave DNA. So PCR is not used for cleaving DNA. Instead, restriction endonucleases are the enzymes which are used for cleaving DNA. So our final answer for this question is option 3, that is DNA amplification. So DNA amplification is utilized for PCR. Hope you understood this well. Best of luck. Thank you.